What is Truth? Darwinism or Intelligent Design? By Mark Lamontia. Mark Lamontia is a mechanical engineer, scientist, and apologetic speaker who regularly addresses questions at the intersection of science, faith, and worldview. This talk was presented at Answering the Big Questions, an event hosted by Apologetics for the Church at Ephrata Community Church on March 7th, 2026. You can find a list of references for this talk at the end of the video and in the description.
Yeah, hi everybody. So thanks everybody for coming. All you guys that love the Lord with all your mind, you get 100 points. So that's good. So this is called What is Truth? Darwinism or Intelligent Design. And I'm attempting here to show you that by science, you can know for sure that God is the creator. I've given this talk a lot of time. And in our culture, we're dominated by Darwinism. Darwin published The Origin of Species in 1859, and he said all the species that ever lived, all the plants and animals evolved by natural selection, acting on random variation. And so God was permanently unemployed. He never had to do anything. And so as a result of that, at universities, God died, and Charles Darwin first the murder weapon. So perhaps as Christians, we should look at what evolution can explain. And we're not going to do that by the Bible. We're going to do that by mainstream science. So we can argue this point by mainstream science. I think a very good case can be made. I'll try to make it that science is the best reason to believe in God. And so the first thing about evolution is don't ever answer a question with the word evolution in it, because it'll change definition 6 times in the sentence. And we're going to look at the 6 definitions of evolution. And the conclusion before the slide is evolution is true for definitions 1 through 3, but it's not very interesting because it holds no philosophical weight. And where it does hold philosophical weight in evolution definitions 4, 5, and 6, it's not true at all. Okay, so that's where I'm going. So definition number 1, change over time. Do you believe that there are different things living in the past that are living now? I do. So then you believe in change over time. You are an evolutionist. But that's trivial, right? That's trivial. And agree to this, and later we will tell you more. And this is the strategy that evolutionists use to get people to lose their faith. They agree with something trivial and then they expand it, right? So change over time is true, but it's not very interesting. So what about definition number 2? And I usually get a aw on this slide, right? And this is a Photoshop slide because you got the bunny rabbits on the left, and if this was true, probably they'd be eaten and all that. But anyway, definition number 2 is change in allele frequencies in a population. Allele is just something that makes some dogs brown and some white, some rabbits black, some white, right? One of several forms of a gene that arise by mutation are found at the same place on a chromosome. And the best definition of this is the peppered moth. So here in the Midlands of England in the middle of the 19th century, they found this moth called Biston bitularia, and it comes in 2 versions. At the top, the white moth, and the black, the melanic or black moth. And the idea by this scientist named Kedawal said, when the trees were darkened by industrial smoke, you couldn't see the dark moths very well on the trees, so the birds ate the white ones. But when pollution laws came around, the trees were lightened up again, and it was easier to see the black moths, and they had the advantage, and the white moths had the advantage. So you had a change in the ratio of light and dark colored moths. Now, do you believe in the possibility of a change in the ratio of light and dark colored moths? Then you believe in evolution, right? Now, notice something. In this experiment, no moth changed color. This is not an explanation of the appearance of color of wings or of moth wings or the moth. Big deal. So it's true, change in allele frequencies in a population, but it's not very interesting. So how about this? You hear about this in the news, microevolution, right? There's Charles Darwin. He found all these moths on the Galapagos Islands in the 1830s. He brought them back and he noticed that some of them had small beaks, but when you had drought years and you had the last tough seeds on the island, some of them had big beaks, and this is called microevolution, right? Microevolution is the change in the size, shape, or color of an already created organ. All right? It happens. It happens. So Darwin wrote this book called The Origin of Species. So I photoshopped his title and I said, look, if he would have called his book How Already Existing Species Undergo Minor Changes, nobody would be arguing about this, right? Nobody. Now, does microevolution happen? Do you believe in microevolution? You ought to. You look different than your parents. Microevolution happens, but it happens by a process of devolution. If you want to read a really good book on this, there's one by Michael Behe, and he shows that all this stuff happens by genes getting broken, not created. Now, I Googled examples of Darwinism, and it's very interesting that all of the examples they show are either microevolution or cartoons. For example, they show microevolution of finch beaks. They show microevolution of the lynx of giraffes' necks. Or they show cartoons of four-footed land creatures turning into a whale. We'll talk about that later, right? But cartoons showing the change in ratios of already existing long and short-necked giraffes or long and small beak finches do not show how you get a neck in the first place, or a giraffe, or a beak, or a bird from nothing. So microevolution is true, but it's trivial. It's not very interesting, and it's not innovative at all. You don't get hearts and livers and kidneys and legs and brains from nothing by microevolution. And it's also misnamed. It ought to be called adaptive variation because nature does not, by selection, select anything. Nature just kills animals, some faster than others. All right, now let's get to the interesting one, speciation. Speciation. So you got these flies, right? Great picture. And a couple decades ago, Jonathan Widener wrote this book called The Beak of the Finch. And he won the Pulitzer Prize. And it says this, the origin of species says very little about the origin of species. That's interesting, right? The book, The Origin of Species, does not document the origin of any species or a single case of natural selection. I came up with this slide, took me a long time to do, all right? So this is a cool slide. This is good PowerPoint, all right? This is an example of what you find in the natural selection literature about speciation, separated breeding groups. So you've got a bunch of fish, right? And you've got change in geography. And they'd be able to separate in separated breeding groups. And after a while, they get separated and they can no longer reproduce with each other. So what? Where's the genuine innovation? That's speciation. Who cares, right? Who cares? Separating breeding groups do not explain the arrival of new complex organs or new organisms. So here is in the literature an example of what you read about speciation. Now, this is a gross slide, so I hope you can keep your, breakfast in your stomach, all right? So this is what they find. This is what's published. Some flies have more hair on their legs than others. Wow. Some flies lick themselves more often than others. Aren't you glad you heard that today, right? Some flies prefer flying up when they take off. Others prefer flying down. Now, these are experiments in the lab. They separate the breeding groups. This isn't in nature, right? And they repeat this separation for generation. Then they regroup them together and they say, I'm going to publish a technical paper because we found incipient isolation. Now, what's incipient isolation? That's a weasel word saying beginning to exist. And if I waited for 1 billion years, we'd have big changes. That's what they say. That's just a weasel word, right? Now, you never get complete isolation, which was what would have to have happened to demonstrate a new species, and you never get large-scale morphological change. Now, if you're in the technical area, you want to publish your results in journals. That's how science works. And the most famous journals in the world are Science and Nature, and big-time journals in the area of evolutionary biology are the Journal of Evolutionary Biology and the Proceedings of the National Academy of Science. If you get your papers in these journals, big deal. Now, here's a quiz. Let's see if you guys can get it. How many confirmed cases of genuine speciation, creating new families or genuine new features, are there in the technical literature?
0.
0. That's amazing. Everybody says evolution's true. And if we look in the right books, if we look in the right journals, we'll find the evidence where it's true. Go find it. You can't find it. It's amazing. You've got this theory, and there is no evidence for it in the technical literature. How can that be? How can that be? So, in conclusion, speciation would be interesting if it were true, but it's never been observed. It is not documented in the technical literature. Now, let's get to the big ones, right? Universal common descent. Darwin had all these notebooks that I wrote besides the origin of species, and in notebook M, he puts this diagram in here and he writes the word I think, and you can get t-shirts like this, right? This is the tree of life. And this is the tree of life that you see. What does that say? In the beginning was a little bug called Luke, a last universal common ancestor, and then you get a branching tree that grows up to all the species, families, genera, and phyla that we've got in life. And so this is in National Geographic. You can find plenty of these, right, where you start with the last universal common ancestor, then you've got Cnidaria, and you branch up into bugs over here. You've got bears, starfish, humans, all that right over here. That's the tree of life. Is there a tree of life? So let's look at this. Does the fossil record prove that there's a tree of life? Or maybe there's something else, right? So we got to start with animal phyla. Anybody here know what animal phyla is? That's the big divisions, different ways of being alive. So one way is you can be a sponge or a cnidarian or an arthropod or a chordate. Now, all you people here, you have a spinal cord. You're chordates, right? All you guys are chordates. All right, so let's see if this is what happens in the fossil record. So we're going to draw the geological column, right? And in this geological column, we'll have the ages, 800 million years ago, 600 million years ago, 400, 200, and 100, and we'll put all these phyla on this timeline. And you would think the man on the street will say, we started with stuff that was simple, and then we got to jellyfish, trilobites, and fish. We got to more and more complicated. Doesn't that sound like what evolution is? Doesn't that sound? Now, what happens in the fossil record? Is that what you find? Is this what you find? No, that's not what you find. All the phyla show up at the same time. Did you know that? All the phyla show up at the same time 532 million years ago. There's none of this. That's not wrong. Now, look at this technical journal to see if we can prove that, right? Time magazine. Evolution's Big Bang. Does that sound like evolution? What does it say? New discoveries show that life as we know it began in an amazing biological frenzy that changed the planet almost overnight. So Time magazine says you want to believe in evolution, it didn't happen, right? So what does evolution say? You got this bug, last universal common ancestor, and then they split out and they change a little bit. They change a little bit, right? You got diversity right at the beginning. And if they change enough, you got a new genus, right? And they change into something different, you got a different genus. And if you get enough change, you get a new family, right? Then you can get a different genera over here and a different family. That's what Darwinism says. Is that what happened? Is that the data? Get in your car, drive to New York City, go to the New York City Museum of Natural History and walk into this room, and they got this big wall of fossils. Pretty cool, right? And when I went there, I said, I know that in March of 2026, I'm going to be talking to all these people. So all you people, get out of the way so I could take this picture. But they didn't listen to me, right? But they got all these columns, right? Now look at these columns. This is interesting. This is the data. Look, there's diversity within the type. There's diversity within the kinds. Remember in Genesis, it talks about kinds. There's diversity within the kinds, but you don't go kind to kind. You don't go there's no cross-phyla evolution at all. So the picture here is a lawn. There is no tree of life. There's a lawn, right? There's another cool slide, right? Let's put these together. So you get all these phyla. They're all different. There's no evolution. There's diversity within the type, but there's no transitions. There's no transitional forms at all. So here's what Darwinism says. You got diversity, then you got disparity. Here's what happened. You got disparity, then you got diversity. It's completely opposite. Something's got to give. The data does not support the theory at all. And Darwin knew this. If you read The Origin of Species in chapter 10, it says, why, if species have descended from other species by insensibly fine gradations, do we not everywhere see innumerable transitional forms? Let's translate that. Why, if my theory says species evolve, don't we ever see it? Right. He knew it. That's in The Origin of Species, right? Now, sometimes you see these deceptive cartoons, right? We're going to go from this 4-footed land creature and evolve into whale. That's evolution. You see pictures like that, right? I'll show you evolution. There's the Corvette. Now, what do we know about the Corvette? They're all designed. So even if Darwinism was true, we could not say that it was by evolution because it could be equally evidence for intelligent design. You can't tell anything from fossils. And again, Darwin knew this in chapter 10. He says, the case at present must remain inexplicable and may truly be urged as a valid argument against the views here entertained. Now, if you look in the fossil record, what happens? Do you see evolutionary history and then you see species appear? No, they show up without any evolutionary history. I could show 19 examples of this. I'm only going to show a few. Jawed fish, poof. The first one, fully formed, 425 million years ago. Winged insects, poof. They just appear. The very first one, fully formed, right? Land animals, poof. They just appear. Flowering plants, what Darwin said, my abominable mystery. The very first one looks like the ones we got now, right? Placental mammals, poof, they just appear. There's another good slide, right? Evolution of modern birds. We'll have a bird fly there. 65 million years ago, they just appear, right? So whatever Darwin proposed about evolution is not what happened in history. The fossil record is as un-Darwinian as it could possibly be. Now, the lead of the American Museum of Natural History about a generation ago was Niles Aldrich. So listen to what he says, his most famous quote. We paleontologists have said that the history of life supports the interpretation of gradual adaptive change, all the while knowing that it does not. That's amazing. That's amazing, right? Right? Okay. Darwinism. The big one, right? Macroevolution, right? So, what does it take to build a new animal? Darwinism or intelligent design? And so, DNA over there, right? New functions require new code. If you put a new function on your computer, what do you need? You need new code. In animals, if you want to have a new function for an animal, if you want evolution, you have to have new code. Now, this is a picture of the number of cell types versus millions of years before the present. And you start with sponges, 5 cell types. Then you get to arthropods, insects. Then you get to amphibians, and then you get to birds, right? And here's another way to show it. Sponges have 5 cell types. Starfish have 41 cell types. Squid have 73 cell types. Crocodiles, 148 cell types. Humans, you have 210 different cell types. So if you got to get all those cells in your body, you need a lot of new information. You need a lot of new code because the way you get these cells is by having new proteins, right? The proteins get encoded by DNA. All you guys have about 40 trillion cells. All those cells are made of 42 million proteins. Where's the information content upon which to build the proteins? The DNA. So you ought to be interested in this one, all right? Now, viable proteins are rare. Doug Axe, who's part of the Discovery Institute, wrote this paper, and he says, let's estimate the prevalence of protein sequences adopting functional enzymes folds, which basically means if you put amino acids together, how many times do you get a good one that you can use in your body? And how many times do you get junk? Because the evolutionists want to say, we'll get X-rays, in the Marvel movies, the DC universe, we'll get spider bites, we'll get a little accident and we'll have a new function, right? Is that true? Or are most of those mutations pretty lousy? All right. So he found the chance at a successful fold is 1 chance in 10 to the 77. Not very likely. For every DNA sequence that generates a short functional protein fold of only 150 amino acids in length, there are 10 to the 77th power non-functional combinations. Now, an analogy here works well. Let's say you have a paragraph of 100 words, and you scramble the letters. You can scramble the letters 10 to the 77th times before you get the one good one. That happened because you, as an author, made the good one. So you can get 10 to the 7th bad ones that will not fold into a stable 3D protein structure capable of performing a biological function. It can be part of your body. Dean Meyer says the difficulty of a mutational search for a new gene or novel protein fold is equivalent to the difficulty of searching for just one combination on a lock with 10 digits in 77 dials and getting it the first time. Not very likely. So there's no one way to mutate a protein with one mistake at a time to get a new functional proteins. So these viable proteins are rare. What's the conclusion? Proteins are not nearby cousins. If somebody says they can make one little change and get a new protein, forget it. They're like islands separated by galactic oceans. Another thing is, there's this thing called orphan genes. Now, orphan is usually spelled O-R-P-H-A-N. And this is a play off that, orphan genes. The idea is orphan genes are only found in individual organisms. How can that be? Orphan genes have been found in every genome sequence to date, from mosquitoes to man, from roundworm to rat, and their numbers are still growing. If you're going to have evolution, you're going to have evolutionary relationships between species. You're not going to have 28,000 genes that only show up in one organism. You're not going to have that, right? So orphans have no evolutionary history. If they did, you would have found a sequence that would have led up to them. So lots of genes. Orphan genes are exclusive to species or families or genera. Now, this is the cool part, right? Where do proteins come from? Where do new proteins come from? So let's look at a pretty interesting protein that's thought to be present at the origin of life. This is called DNA polymerase III, and this is its shape. And DNA polymerase III is pretty important because it's the thing that splits the DNA into 2 strands and replicates it. And if you didn't have that, you couldn't reproduce. So this is useful in reproduction. Now, the other thing that you find in technical literature is it shows no similarity with other polymerases. Now, to show you DNA polymerase III, I have to teach you one thing that they do in the technical literature. Geneticists get tired of naming all the chemicals: serine, asparagine, glutamine, cytosine, glycine, proline. They get tired of saying those words. So instead of those words, they use letters: S, T, N, Q, C, U, G. There's 20 of these amino acids. They just use the letters. So you're going to see a string of letters. So here is DNA polymerase III. All you have to do is get those 1,159 acids in the right order, and you got it. Great. Now, how do you get those 1,159 amino acids in the right order? Let's look at 8 amino acids. DRISKLIP. 8 amino acids, just those, right? All right. So here's the 8 amino acids, and they end up with aspartic acid, arginine, isoleucine, all the way down to proline. Now, how in the world did those get there? How did your body get those amino acids in that order? By the DNA. The DNA unwinds, and this is the message-bearing axis of the DNA that gets read. That's where the information comes from. The DNA unwinds, right? So I couldn't figure out in PowerPoint how to make the DNA unwind. So I put this movie in there, and you split the thing open, and you expose the nucleic acids, right? You expose the nucleic acids. So here, Now you're inside the nucleus, that's the great part, and outside the nucleus is the cytoplasm. So you split the thing in half and you read it and make an mRNA transcript, and then you go outside the cell, and now you're going to try and line up those amino acids. So the first three are CUG. Now, if you know the triplet code, C goes with G. There's G. U goes with A, and G goes with C. So that would be ASP, right? Aspartic acid. So we just did DNA protein synthesis right there, right there. Now that is computer programming. That is computer programming. Now I want to stop a minute. Look at how smart God is. All we do is figure out how it works. He thought it up. God is really smart, okay? Really smart. Okay, let's do another one, right? UCU. You think we can do that one? Right. U goes with A, right? C goes with G. U goes with A. Arginine, right? DNA protein synthesis. Your body is doing this 1,000 of these per second in every cell right now, right? This is what's happening. So Bill Gates, right? He heads up Microsoft. He says this: DNA is like a computer program, but far, far more advanced than any software we've ever created. Smart guy. And so if you keep on doing this, you get this string, and it's 8 here on the screen, but it's really 1,159 amino acids long. All right, now, what's the chances of getting this by chance? What's the chances? Now, there's 20 proteinaceous amino acids, so what's the chance of getting aspartic acid? 1 chance in 20. 1 chance in 20. What's the chance of getting arginine? 1 chance in 20. What's the chance of getting the rest of them? Every one, 1 chance in 20. So what's the chance of getting the whole thing, right? 1 chance in 20 to the 1159th power. That's a big number. If you use logarithms to convert that to base 10, that's 1 chance in 7.7 times 10 to the 1507 power. That is a big number. How big is it? How big is it? To get one number on a dice, it's 1 chance in 6. To get 2 6s, it's 1 chance in 36. To get twins, it's 1 chance in 67. To get a bicycle lock with 4 dials on number 10, it's 1 chance in 10 to the 1,000. To get hit by lightning, 1 chance in 960,000. To win the Powerball, 1 chance in 292 million. There's been 4.3 times 10 to the 17th seconds since the Big Bang. The number of elementary particles in the universe is 10 to the 80th. The number of events that have happened for all time is 10 to the 141st. The universal probability bound is 10 to the 150th. And to get a 1159 amino acid sequence protein by chance is 1 chance in 7.7 times 10 to the 1507th, which is why in the technical literature, everybody says you can't get it by chance. Forget it. It can't happen. So, where do new proteins come from? And the marching band problem. You got to get the protein once it's made. It's got to go to the right place in your body, right? So I'm going to show the marching band problem. Now, you can maybe guess where I went to college. This, Ohio State, this is as close to heaven as you can get while you're on the earth, right? So I go there with my wife once a year, every year, right? And every game they play Script Ohio. And the band marches around, they play Script Ohio. But before, if you go to the skull session, they have the band sitting down and they play Script Ohio. So let's see what the volume's like. All right, nothing like hearing Buckeyes music. Okay, so what is the difference between these guys playing the song and these guys playing the song? These guys know where to go. That's information based. They have information where to go. Where does information come from? It comes from authors. Who is the only author available at the time? Only one guy around, right? Right? Only one guy. So this is body plan morphogenesis. That's an interesting word, right? Morphogenesis. You're going to try and generate the morphology. It is a massively information-driven process. So that's called the marching band problem. Let's talk about it. Do scientists know how you go from DNA to proteins, to organelles, to cells, to tissues, to organs, to zygotes, to fetuses, to a human? Do they know that? Do they know that? No. 1953, Crick and Watson, they figured out how do you go from DNA to proteins. That's all they know. Do they know the rest of it? They don't know. They don't know. Nobody knows. Nobody knows where the information content comes from to get the rest of it. Nobody knows where you get the blueprint for the organism. Nobody knows the assembly instructions, and nobody knows the tools. And you cannot even put together a tricycle on Christmas Eve for your kid without all those things. You can't do it, right? Now, let's see if I can prove to you that DNA is not the only thing. This is Lego Creator. Lego Creator. With the Legos in this box, I can make a monkey, right? I can make a squirrel. I can make a parrot, all with the same Legos. DNA, what does it do? It gives you the parts, but it doesn't tell you how to put together. DNA just gives you the parts. There's missing universes of information that biology has no idea where they come from, as shown by this Lego. So this is no different than a house. In a house, you got to have all the materials. That's like the DNA. That's good, right? But you also need a blueprint. You also need to know how to put them together in the assembly instructions. Doesn't do very good if you start with the roof and then put the foundation on later. You better do that right, and you got to have the tools. Now, all these things right here, those are information-based. Information comes from authors. When science finally figures out where the blueprint is, where the assembly instructions, and where the tools are, they will find that those are information-based things. Information comes from an author. And there's only one author around. That's called God. He's the only guy that was there to do it, right? Right? So if you see information, you are seeing God. You are seeing God. Now, there's another problem with evolution, embryonic lethals. So you got DNA, and then you got to fertilize an egg, and you start making a zygote, and then you got a fetus, and finally get a baby. Yeah, keep it. Nobody. You want to go ahead and you want to change this animal. How are you going to do it? Right? You better make a mutation up here. If you make a mutation up here, all you're going to do is give the baby a scar, right? You better make a mutation here if you want to have evolution. But the trouble is mutations here kill the fetus. So it doesn't work, right? Mutations here only affect a few cells. The body plan is also already established. If you want to make a change, you got to make it early. But if you make it early, you kill it, right? Now, there's a similar one here. With a complicated name. This is a very easy concept, but a very complicated name. Developmental gene regulatory networks. All this says is you got to have A before B and B before C and C before D and D before F and all that. You got to have things in the right order. And there's a scientist that figured this out. He just died 2 years ago, and he called it developmental gene regulatory networks, and that gets everything in the right order. Right? If you want a new animal, you have to have a new developmental gene regulatory network, right? But Eric Davidson, the guy that figured this out, says mutations in the developmental gene regulatory network lead to catastrophic loss of the body part or loss of viability altogether. What does loss of viability mean? Loss of viability mean? It means you're dead. You're dead. You need to change that to get evolution, but you can't change it because if you do change it, you're dead. All right. There's always an observable consequence if the developmental gene regulatory network is interrupted. It's always catastrophically bad. So if I were to tell you, I can tell you how to get from a wolf to a German Shepherd, you'll probably agree with me. But if I say you can get from a 4-footed land creature to a whale, man, you need to make some big changes. So it isn't going to happen. It isn't going to happen, right? Now, what about multi-part systems? We're going to give Darwinism all the single-part systems that can make one change and that gets selected. Are there any of those in life? No, there aren't any. So let's look at some examples of these, right? So Darwin said, this is his failure criteria. If it could be demonstrated that any complex organ existed which could not possibly be informed by numerous successive slight modifications, my theory would absolutely break down. Why did he say that? He wanted numerous successive slight modifications so he could get God out of the picture, right? If you could have that, you can have little accidents and then you could have evolution. He wants to get God out of the picture. And the example is a mousetrap. You got to have all the parts for the thing to work, right? So a generation ago, Stephen Jay Gould, who was the lead paleontologist in the U.S., and Richard Dawkins were having an argument. And Gould says, what good is 5% of an eye? And Dawkins says, I don't think that's a very good question, right? So let's say you're this 3D man that you get off of the internet, 3D man. He can't see. He can't see, right? He wants an eye. So you give him an eye in this present. What does he do? Throws it in the trash can. Doesn't do him any good because vision is a multi-part system. You see that? It doesn't matter if you get one part because Darwinism would have to select it every single time for an advantage, so that doesn't help you. So let's look in life. Molecular motors. So here's a bacteria, and they have this little flagellum motor that they use to swim around looking for food. That's the bacterial flagellum. Does that look like that evolved? Does that look like a machine? That doesn't. It's not like a machine. That is a machine. It's got bearings and rotors and gears and a little propeller on the end to allow the bacteria to go ahead and move around, right? There's another picture of it, right?
Resembles a human design move.
It's a basically an outboard motor. I'll skip this part. Here are kinesis. This is running around in your cells right now. Isn't that gross, right? It's carrying cargo vesicles around in your cell to get rid of all the junk, right? That's a molecular motor. That's a walking protein, takes 4 parts, not one part at a time. If you could make that one part at a time, Darwinism could have made that, but you can't because it takes 4 parts. Now, this is very similar to going, this is doing what this is doing. Did this evolve or did somebody design this thing? Right? All right. Now, there's my wife's favorite one. This is ATP synthase. This is the mitochondria. The ATP synthase is how you get energy. You guys are sitting there breathing right now because each of your mitochondria has 50,000 of these ATP synthase, which is a turbine that creates ATP from ADP. That's how you get energy. That's a multi-part system. It cannot possibly evolve by Darwinian evolution because it's got so many parts. How about cascades? Cascades cannot be made by the Darwinian mechanism because there's not anything to select until the end. And the best example is blood clotting. If you cut yourself and you don't have a clot, all the blood isn't going to come out. That's bad, right? You can't have that. So let's look at the blood clotting system. So there you got the red blood cells and they're moving in the blood vessels, right? And you got a wound, right? And the blood starts dripping out. That's bad, right? So down here, you want to get this clot. So the first thing that happens, you get Hageman factor, which combines with HMK to make activated Hageman factor. And Darwinian evolution selects that, right? Because you got a clot, right? No, because you don't have a clot. All you have is the first thing. You don't have a clot yet, so Darwinian evolution cannot select it, right? So then Hageman factor activates prekallikrein into kallikrein. Does Darwinism select that? No, because there's no function to select. There's no function. So then you go ahead and you do all the 19 functions so that you finally get a clot only when you need it, only where you need it. That would be really bad if you got a clot all the time because then you would freeze up solid, right? Or you would bleed out, right? So you have to have all these 19 steps. Now, when I had the AFib operation, I went to the doctor and he's about ready to do an operation on me. I asked him about all this stuff. He said, where did you learn all this stuff? Right? So cascade, you cannot make a cascade with Darwinian evolution because you got multiple steps. Darwinism gets all the systems in your body with one step. There aren't any. How about this one? Now, I hope you guys like this, right? All right? Hope you guys like this. Everybody hold your breath, put your seatbelt on. This is the best data there is. This is the male reproductive system and the female reproductive system, and nobody likes to look at these on the slide, so we'll close the curtain. But these are systems on 2 organisms. So we'll open the curtain again and say the female reproductive system has 10 parts, and the male reproductive system has 14 parts. So all you have to do to get Darwinian evolution to work is, let's say you got the female reproductive system, unless you have the evolution of a male reproductive system at the same location on the planet at the same time. It doesn't matter that you got this, right? Because it doesn't do any good without this one. You think evolution did that? You're crazy. It didn't do it, right? And then, so general multi-part systems. Darwin wrote a book called The Origin of Species because he knew what life looked like, but he didn't know how it worked. If he knew how it worked, he would have never written that book. And Steve Loftman and Howard Glicksman, who are both on the engineering research group of the Discovering Institute with me, wrote the book called Your Design Body. And they say, if we look how life works, the case is closed. Darwinism fails miserably, design is required. And so their best example of this is how you get oxygen to every cell. Now, if you were an amoeba, it'd be pretty easy because oxygen's right outside your membrane. But if you're a human, almost all your cells are on the inside. So your skin cells can get the oxygen, but the cells on the inside, they can't do it, right? So all you need to have is you need to have a respiratory system and a gastrointestinal system and a circulatory system and a way to get the CO2 out of your body, et cetera, et cetera, et cetera, just to get oxygen to every cell in your body. And if you think you don't need that, hold your breath, and it won't be too long till the respiratory system in the back of your neck says, you better breathe, man. You better breathe, and you need to have all this stuff. And these are all the problems have to get solved just to get oxygen to every cell. You cannot do that with Darwinian evolution. It's impossible because it's multi-step. So the conclusion of this talk is evolution true? Sure. But where it's true, it's not very interesting. And where it's interesting, it's not true. Right? So the conclusion of the talk, there is no evidence for macroevolution. The fossil record is about as un-Darwinian as it could possibly be. Darwinism faces numerous in-principle roadblocks: the information content in the cell, the information required for body plans and assembly, irreducible complexity, and multi-part systems. What does the word in principle mean? In principle means impossible. Darwinism isn't found to be true now, and with 500 more years of National Institute funding, it will not be found to be possible. It is impossible in principle. It won't work. All are markers for intelligent design. Darwinism makes a weak and limited argument by considering how life looks. If we look at how life works, Darwinism fails miserably. There's 6 definitions of evolution. Where evolution is true, it's not very interesting. Where it's interesting, it's not true. And shifting the definition of the word evolution is the tactic used by materialists used to win arguments. So if somebody says, if you believe in evolution, say, define what you mean by evolution. The scientific evidence is on the side of intelligent design. Darwinism does not need to be improved. It needs to be replaced. Now, did God do it? When you see cascading problems in life, when you see feedback control in organisms, when you see multi-part systems, when you see coherent systems that are interdependent, you should conclude you are witnessing the fingerprints of the intelligent designer. There's only one candidate, and that's God. So there you go. What is truth? Intelligent design is truth. So I hope you enjoyed that.
If you found this talk helpful, leave us a comment and like and subscribe so you can stay updated on future content. To learn more about our ministry and access additional resources, visit apologeticsforthechurch.org. Thanks for listening.